Good morning to all of you. So, today I am going to discuss with you all about a new chapter that is given on your recombinant DNA technology subject. And first, I'm going to discuss about the what is recombinant DNA technology and how what are the tools required for recombinant DNA technology experiments. So, what happened in 1973? Two scientists named Cohen and Boyer. So, two scientists named Cohen and Boyer produced the first recombinant DNA by adding an antibiotic resistant gene from taken from the Salmonella typhimurium. So, from Salmonella typhimurium, typhimurium, they have isolated a gene which is antibiotic resistant, which has the property of antibiotic resistant. It has this antibiotic resistant property, and that gene is inserted into E. coli cells. That gene has been inserted into the E. coli cell via plasmids. Now, I think you all know, you have studied in your microbiology class what is plasmid. Plasmid are the extra chromosomal double stranded circular DNA molecules which are found on the prokaryotic cell that are capable of independent replication, and they carry some genes which are carrying that antibiotic resistant property, okay, needed for conjugation to transfer into from one bacterial cell to another bacterial cell. So via plasmid, they have inserted that gene into the E. coli cells and expressed that gene. So this is what recombinant DNA technology means actually. So in what is recombinant DNA technology or technique? In recombinant DNA technique, a specific DNA segment is isolated from specific cell, and that DNA segment that is isolated, it may be carrying multiple genes or a single gene coding for a particular protein or enzyme. That DNA segment which has been isolated, it is then integrated, that means inserted into another specific cell, so that the new DNA segment is obtained. The new DNA so obtained is called your recombinant DNA, and this technique is known as the recombinant DNA technology, or simply called as RDT or genetic engineering. So here you can see I am zooming in now. So here it is the donor DNA. There was specific organism. Let's suppose there is a organism like um, mammalian cell. Suppose mammalian cell is there, and from mammalian cell we have isolated the DNA. There are various ways through which we can isolate the DNA, like phenylchloroform extraction technique. Then we cleave the DNA, we cut the DNA to get our specific DNA of interest by using restriction enzymes called as restriction endonuclease enzymes. Restriction endonuclease enzymes, you will see in detail later on, they actually identify specific sequence in the DNA and cleave that sequence, cut that sequence. Thus, we get our desired DNA piece that we want to insert. We need some carrier molecules to carry this desired DNA, and that's why we use plasmid DNA. Plasmids are extra chromosomal double stranded circular DNA molecules which are found in prokaryotic cells that are capable of independent replication. So, with the help of same restriction enzymes, we also cleave the plasmid DNA as well. So, we have cleaved it. After cleaving it, what we have done, we insert this DNA of our interest, carrying that desirable protein into that plasmid by the help of ligase enzyme, and thus we produce a recombinant DNA. Now this recombinant DNA is then introduced to the host cell where this plasmid will makes millions and millions of copies, and thus our gene of interest will get millions of copies will form. Okay, sometimes what happen if our main aim is to express this gene, then we modify this plasmid, we give some other sequences, regulation sequences which are needed for transcription of that particular gene segment to get our final protein product. So this can be also be done, but for that we need some specific arrangement, special arrangement into that plasmid. Okay, so we introduce them into the host cells, and after introducing them into the host cells, multiplication of the host cell occur, and then we will select selection of the clones will be done. Now selection of the clones can be done by using various ways, such as the genes which has been used in the plasmid um, may carry some antibiotic resistant gene property or some other property which will emit light, like for example the green fluorescent protein, which are normally introduced into the plasmid for proper selection process, or the blue white screening we will see in the PUC vector, where specifically we have inserted the laxate prime gene, okay, where it interacts with the beta where it interacts with the beta galactosidase to create a blue product. Okay, so in this way we get that which colony of the E. coli cells or bacterial cells or whatever organism we use. We can use a mammalian cell line also. We can use a mammalian cell line like Chinese hamster. Chinese hamster is actually look like a pig, guinea pig. Okay, it looks just like a pig, guinea pig, and nowadays Chinese hamster cell lines are also being prepared, used as a host cell. Okay, and thus when we get our desirable product, then what we do, then we isolate that product known as downstream processing, and carefully by various purification techniques like chromatography techniques. Okay, and then we commercialize them, we stabilize them and commercialize them in the market. For example, the insulin synthesis gene. Okay, insulin is very much required for the diabetes patients who don't able to make sufficient insulin, thus increase their blood sugar levels. So like for example, insulin synthesis gene, it is isolated from the beta cell of pancreas. So from the beta cell of pancreas we isolate the insulin gene, suppose. So I'm drawing here. So from the beta cell of pancreas we isolate the insulin gene. That means the gene which will uh, expressed to form insulin protein, and this gene is now added to the plasmid of E. coli. So it is inserted into the plasmid of E. coli, suppose, and then this plasmid is allowed to introduced into the host cell. Normally we use host cell as E. coli cell because as we have taken the plasmid from E. coli, so it can well adapted to insert them into the E. coli cell, and thus we multiply the plasmid into the E. coli cell to get more and more insulin gene copied. And if our aim is to get that product, we add some specific regions into the plasmid so that the insulin gene also get transcribed and translated to form that protein. Further, with the help of downstream processing, we isolate that protein product from that cell. In this way, your recombinant DNA technology or genetic engineering process is carried out. So, what is the full definition of your recombinant DNA technology? Recombinant DNA technology is the process by which a DNA molecule can be isolated from a specific cell and then can be inserted in another DNA molecule at desirable position to obtain our recombinant DNA, that is the modified DNA. So again I repeat, recombinant DNA technology is a process by which a DNA molecule can be isolated from a specific cell and then it can be inserted in another DNA molecule at our desirable position to obtain recombinant DNA. Okay, now why this type of recombinant DNA molecules are being produced? It is produced due to many reasons. The first reason is your to obtain large number of copies of that specific DNA fragment. So to obtain large number of copies of our desired DNA fragment. Okay. Second important thing is to recover large quantities of protein produced by that concerned gene. So our second important reason that why we are making this recombinant DNA molecule is to recover large quantities of our desired protein, which is produced from that our desired gene by that concerned gene. And the third second point is to recover large quantities of the protein produced from desirable gene. Okay, and the third important point is to insert the gene, to integrate the gene into the target organism where it will express itself. Now for doing this, for doing all this stuff, we need a specific vector. We need a specific vector, and generally we use plasmid as a vector. So here we can see is simplified. I have drawn the plasmid in the form of circle. So for achieving this, it is first important to isolate the desirable gene and insert that gene segment or segments if you are wanting to more insert your gene of interest into a suitable vector. Now here the plasmids are the vector molecules. What is vector actually? Vectors are the carrier molecules. Just like we also say mosquito fly as a vector. because they carry away the pathogenic organisms that cause malaria, dengue, etc. Similarly, here the plasmids are also acting as vector because they are carrying the desirable gene of our interest into suitable host cell. So you can see here this plasmid is acting as a vector, and this plasmid is carrying our DNA of interest into the suitable host cell. Here the host cell has been shown as E. coli cell. We can also introduce them in mammalian cell. For that we need some modifications in this vector. That's why we create mammalian artificial chromosome. We also create yeast artificial chromosome known as yak factor, mac factor. Okay, so that it can be expressed into the mammalian cell. But for the simple vectors like PBR322, you hear this name before in your class 12 class. PBR322, this actually is being assembled. It is a vector, plasmid vector, which has been obtained from E. coli cell and has been assembled. Okay, so most commonly used vectors are the bacterial plasmids. Sometimes we can also use bacteriophage. Bacteriophage can also be used as a vector. Okay, they can also be used as a vector. Virus can also be used as a vector, like the potato mosaic virus, the tumor TMV, tumor mosaic virus can be used as vector. In plants, most of the case the vectors that are used is Ti plasmid and Ri plasmid. I think I have said this name in your first class in your first semester. Ti plasmid and Ri plasmid. These plasmids are taken from a specific harmful bacteria called Agrobacterium tumefaciens and Agrobacterium rhizogenes. So from this bacterium, Ti plasmid, Ri plasmid is obtained, and these plasmids are a little bit modified to be used them as a vector so that we can insert our gene of interest into this vector, carried out our replication process, expression process, and we can able to modify our host cell. Okay, or to get our desirable product maybe. So the desired DNA fragment is inserted to the vectors, and it can be inserted with the help of some enzymes obviously, because to because here we can, I have already said, we use restriction endonuclease to cleave a portion of DNA of our interest, and with the help of same restriction endonuclease we cleave, we cut that same position of the DNA to create that type of sticky ends, so that our DNA can easily get inserted into this vector. So here we use enzymes for inserting our DNA fragment into the vector. Uh, Ligase enzyme is also used to stick that, to join that ends. So here various enzymes are obviously used um, to insert our desired DNA fragment into the vectors, and then the vectors are now introduced to the suitable organism. Here it is showing the E. coli cell, usually a bacterium. Now this is known as host, where we are introducing our desirable DNA, the vector, the recombinant vector, into the host cell. Now in case of E. coli, it is very easily done, known as transformation. It can be easily done by using calcium chloride treatment. Okay, and these transform cells are then selected, they are cultured, and how they are selected, actually these plasmid also carry some other specific genes like antibiotic resistant gene, okay, or some other chemicals which will interact with the other synthetic chemicals to create a colored product or fluorescent product. Okay, that will easily helps us to select that which colony of cells has our desirable gene inserted. Nowadays, there is a big problem that occurs that we normally use E. coli cell. In all the diagrams, you will see in your sixth semester also, you will see that the genes that we have inserted into the vector, okay, that may not get expressed properly due to many reasons, as we have studied very well in our molecular biology classes in translation, that there is a term called post translational modification. Post translational modification. Now this type of modification occurs in the eukaryotic cells, not on the prokaryotic cells. Now suppose you have used the gene taken from the eukaryotic cells in order to express, and that gene is inserted to the vector. Here we use the plasmid. Plasmid is obviously taken from the bacterial cell, and that vector containing our desirable DNA is inserted to the host cell, suppose E. coli cell. So what will happen? Bacterial cell cannot undergo this type of post-translational modification process. That means during post-translational modification, what happened we already know. The change in the structure, conformation, or stability of the protein is achieved. The primary structure is then converted into secondary structure by different hydrophobic interactions, then tertiary structure and quaternary structure. The alpha helix, beta pleated sheet, beta turn are all observed in the secondary structure of the proteins that we know. This type of events cannot be occurred in the prokaryotic cells, since prokaryotic cells does not undergo this type of modification process. So what to do? So since the host machinery is different from the DNA from where we have isolated, so nowadays what we do, we use a suitable cell line specially designed actually for for the gene which is taken from that particular organism. Like if we are if we are wanting to express our human gene, then that gene is cultured in the mammalian cell line. For example, we use Chinese hamster ovary cell line or CHO cell line to be easily cultured, to be used as a host cell. Okay. However, DNA replication is achieved when we can get millions of our desirable gene segments from the colony of transformed cells. So, this was about the overview of your genetic engineering. So, here you can see that this is the basic techniques, basic process which is involved during genetic engineering. Okay, what it is saying, let us see. First, generation of DNA fragments. We create a gene, we have isolated the DNA, and then we create fragments of DNA and selection of our desirable piece of DNA. For suppose insulin gene from beta cell of pancreas. Then in one next step, insert that selected DNA into a cloning vector. The vector, cloning vector means the vector which will replicate continuously. So if we can able to insert our desirable DNA into this type of vector, our desirable DNA will also make millions and millions of clones. And we create a recombinant DNA or chimeric DNA. Now what is chimeric DNA? Chimeric DNA means uh, the DNA containing segments of many fragments actually. Don't say fragments, it is assembled actually, consists of various type of gene segments contained within the vector. Now chimera comes from the monster in Greek mythology that has a lion head, goat body and serpent's tail. The same thing can be observed in our Hindu mythology as well, the Narsimha avatar, in incarnation of Narsimha, one of the Vishnu's incarnation in Indian mythology. So chimera just means different segments. Okay, suppose the antibody resistant gene, you get a desirable gene fragment, then the fragment of the plasmid, then the origin of replication from another source. So you use the origin of replication so that when you are inserting them into the host cell, that particular origin of replication is needed. Then we use another segment of antibody resistant gene, and so on. So we make a chimera in this way. Okay, so we assemble actually, okay, that cloning vector. You will see later on when we will discuss about the individually, we will discuss about the each and every plasmids in separate videos. Okay, next, introduction of the recombinant vectors into the host cell. We will now introduce that vector into the host cell. Like for example, we introduce them into bacteria. Suppose for example, it is very easy to insert them into bacterial cell by simple treatment like calcium chloride treatment or polyethylene glycol treatment. This treatment I have already discussed with you in your plant tissue culture, where protoplast fusion can be done. Or you can also use another treatment like, um, the name of the treatment I have forgotten, but we use electric impulses, electroporation. Yes, electroporation treatment we also use. Short medium pulses are applied for a few microseconds so that the DNA of our interest can get inserted into the host cell. Then multiplication and selection of the clones containing the recombinant molecules. For selection, depending on what type of gene arrangement you have inserted into the plasmid, whether the plasmid have naturally antibiotic resistant gene or not, with the help of that thing we can select our desirable clones containing the recombinant DNA molecules. For example, in case of PBR322, in case of PBR322, um, if this is the PBR322, it consists of two antibiotic resistance gene. It consists of two antibiotic resistant gene. One is known as tetracycline resistant gene, and another one is your ampicillin resistant gene. So it has these two antibody resistant gene. So what happened, we try to insert our gene, and it also has a origin of replication. Obviously it is needed. Origin of replication is obviously is needed, because otherwise how DNA replication will occur in the plasmid. What we do, our gene of interest, say we use black color here, not black color, okay, we use violet color. So our gene of interest is allowed to insert in any one of these antibiotic resistance gene to become inactivated. So this is our gene of interest. We insert them, suppose, into this TETR. Now this TETR becomes ineffective because we have disrupted the gene, but the ampicillin resistant gene is still active. Now what happen, if we when during selection process, if we allow these plasmids, if we allow this bacterial cell, when these plasmids are inserted into bacterial host cell and cultured for certain generations, and when these cultured cells are grown on tetracycline medium, they will not survive, because they have this ampicillin resistance gene present, not the tetracycline resistant gene. Because in this tetracycline resistant gene, we have inserted our desirable DNA. But if we culture the cells in ampicillin medium, they will survive because they have this gene active still now. Okay, so based on this property, we can able to select our desirable gene. select our desirable colony of cells that contain our desirable plasmids having our desirable gene. Now this whole concept is called insertional inactivation, because we have insert our desirable gene to inactivate a particular gene carrying antibiotic resistance property. Okay. And last, expression of the gene to produce our desirable product. So if our main aim is to express that gene, for that we have to make certain modifications in that plasmid, in that vector where we have inserted the gene. Like we have to place the termination sequence, we have to place a region where the RNA polymers will grab on, will bind and carry out the transcription process. Okay, so that to form that RNA, and that RNA will be used for translation process to create the protein, and we get our desirable product within the host cell. Finally, the desirable product will be obtained by downstream processing techniques. Now the main thing is that what are the tools which are required for our genetic engineering process? What are the tools? So what are the things which are needed for genetic engineering? The first and foremost important tool, three important tools are mainly required. If I broadly speak, wait for a minute. So broadly speaking, three tools are mainly required. What are that three tools? The first tool that is required is set of enzymes, assortment of enzymes like restriction endonuclease enzyme, like ligase enzyme, nuclease enzyme, okay, DNA polymerase enzyme, okay, then alkaline phosphatase enzyme. Different types of enzymes are needed for your recombinant DNA experiments. Okay, even to isolate the DNA, we need cellulase. If you are isolating the DNA from the plant cell, pectinase, different type of enzymes are needed. So the first one foremost thing, we need enzymes for isolation of our DNA, for cleaving the DNA. Cleaving means to cut the DNA at specific region which have our gene of interest. Okay, we need ligase enzyme, we need nuclease enzyme, polymerase enzyme for amplification. You will see in the PCR reaction, they don't need any vector, they need only the primer and our desirable DNA and nucleotides to carry out the whole replication process of our desirable DNA. Second thing that is required for as a tool is your vector. Well, vector is obviously it is required. Vector is the carrier molecule. It is carrying our desirable DNA at specific position in the host cell. And what type of vectors we can use? We can use plasmids, which are found in the bacterial cell. We can modify these plasmids into cosmids, into artificial chromosomes like mammalian artificial chromosome, yeast artificial chromosome, human artificial chromosome, and so on. We can also use bacteriophage, like we can use bacteriophage as a vector, like for example the lambda phage, then M13 phage can be used. Lambda phage, M13 phage can be used as the vector molecules, okay, for carrying our desirable DNA. Okay, and the third thing that is required is, obviously we know, host cell. What type of host cell, what type of DNA we are wanting to express? We are wanting to express our mammalian gene, so we need a mammalian host cell like Chinese hamster. Ovary cell line should be used. If you have simple gene product which will produce simple gene, like for instance your growth hormone, then we can use E. coli as host cell or yeast as a host cell. E. coli as a host cell can be used, yeast as a host cell can be used. So suitable host cell will be used for the recombinant DNA technology experiments. Okay, so this was a broadly that I have said that three things are required mainly. So the first one is restriction endonuclease. Restriction endonuclease is also called cutting enzymes. That's why the symbol here in this diagram in this PDF has been used as Caesar, because it is a cutting enzyme. What does it do? Simply, if this is the DNA sequence having 5 prime and 3 prime, it recognize certain sequence. Restriction endonuclease recognize the certain sequence and cut that sequence and cut within that site or nearby that site. That's what restriction endonuclease is meant for. Restriction, endo means inside, nucleus means those who cleave the nucleic acid, DNA or RNA. So restriction endonuclease are one of the important group of enzymes for manipulation of DNA. These are bacterial enzymes that can cut or split the DNA from any source at specific sites. So they recognize specific sequences. I will show you the some specific sequences. You have also learned some specific sequences in class 12, but not too much that was given here. I will try to give you more and more sequence if possible. So they recognize the specific sequence and cut that particular sites. Around that sites they cleave. They were first discovered in E. coli, restricting the replication of bacteriophage by cutting the viral DNA, that is the host E. coli DNA is protected from cleavage by addition of the methyl groups. So firstly, this restriction endonuclease was actually observed in the E. coli cell. What happened? The E. coli cell were used as host cell by the bacteriophage to replicate themselves as a result of transduction, right? So to protect from the bacteriophage, E. coli, what they have done, they use this restriction endonuclease enzyme to cleave the genome of the bacteriophage which has been inserted, and their genome is protected by addition of the methyl groups. So if this is the genome of the E. coli, and if this is the genome of the viral DNA, the bacteriophage viral DNA, okay, then what happened? The E. coli cell have its methyl group already present. Its methyl group is already present, so they are being protected. But the viral DNA is not having the methyl group, so they are cleaved by specific restriction enzymes. They observe specific sequences and cleave that site, making fragments of bacteriophage DNA to make them inactivate. Okay, so thus the enzymes restrict the viral replication known as restriction enzymes. So they are restricting what? These enzymes are restricting the viral replication inside the host cell, here E. coli cell. That's why the name comes restriction endonuclease, because they are restricting the viral DNA replication to occur, and they are endonuclease. That means they cleave, they cut inside the nucleus, inside the nucleic acid. Hundreds and hundreds of restriction endonuclease have been isolated from bacteria, and some of them are commercially available. You can find them in your different websites. You can find different type of restriction endonuclease. Price is little bit high because they are used for research purposes mainly. Okay, not in academics. You can use them in academics, but it is pretty much higher in cost. The progress and growth of biotechnology is unimaginable without the availability of restriction enzymes. Restriction enzymes plays an important role because with the help of restriction enzymes, we can isolate our gene of interest. So if this is the place where we have our desirable gene, so by observing the specific sequences by restriction enzymes, it quickly it quickly cleaves at that site, and thus we get our desirable DNA fragment. right? Now, to name the restriction enzymes, we have a certain nomenclature. Naming them, we have certain nomenclature, just like we for enzymes we have certain nomenclature. So why not with the restriction enzymes? But it's simple. Restriction endonucleases are named by standard procedure with a particular reference from where these restriction enzymes were isolated from, right? The first letter that is given in restriction endonuclease, given in italics mainly, it indicates the genus name of the bacteria from where this enzyme has been isolated, followed by the first two letters of the species. For example, I writing a name of a restriction enzyme commonly used. You all know the name EcoR1. E stands for Escherichia and CO refers to coli. So the same thing has been given. The first letter of the enzyme indicates the genus name and it is followed by the first two letters of the species name, and then comes the strain number, from which strain it has been taken from. It is taken from the R strain, and finally a Roman number is given, eco R1. That 1 refers to the, what is that is it that is this that first enzyme or second enzyme or third enzyme. So eco R1 refers to the first restriction enzyme which has been isolated from the R strain from E. coli organism. Okay, understood this part? So restriction enzymes are named in this way. First capital letter refers to the genus name, followed by the first two letters of the species name. Then comes the strain number, the strain, and the last that is given in Roman number, it indicates the order of discovery. Like here it has been says eco R1, eco R1, E for Escherichia, CO for coli, right? R refers to the, sorry, R refers to the strain number R13 strain, and 1 refers to the first endonuclease enzyme from where it has been isolated, discovered. Then another example, HIND3, HIND, HIND3. H refers to Haemophilus, IN refers to influenza, D refers to the strain number RD strain, and 3 refers to the third restriction endonuclease which has been discovered. In this way, the nomenclature of restriction enzymes are named. Now, different types of restriction endonuclease. At least there are 4 different types of restriction endonuclease that are known. Type 1 restriction endonuclease, type 2 restriction endonuclease, type 3 restriction endonuclease, and type 2S restriction endonuclease. All of the features of these restriction enzymes are same, but little bit there is a difference. What is the difference? Let us see. In type 1 restriction enzyme, what it does, a single enzyme with 3 subunits for recognition. This restriction enzyme is made up of 3 subunits. Okay, they recognize the particular sequence, they do cleavage, they cut, and they methylate that site. Okay, good thing. Where it does the cleavage, that's the main thing. It can cleave up to 1000 base pair from the recognition site. That's a bad thing. Why? Because if this is our DNA and if this is the recognition site for the type 1 restriction enzyme, that type 1 restriction enzyme will recognize and cleave 1000 base pair away, maybe 1000 base pair away, they will cleave. So if our desirable DNA fragment is between this site, then this amount of base pair is pretty huge loss. Okay, it is a loss because so much big fragment of DNA cannot be inserted in some vectors whose molecular whose size is so small, like PBR 322. So type 1 restriction enzyme, they cleave up to 1000 base pair from the recognition site. Next is the type 2 restriction endonuclease, where two different enzymes are there. They cleave or modify the recognition sequence, and the cleavage site is same or close to the recognition site. Now this type 2 restriction endonuclease can be used because they identify the recognition sequence, and cleave means they cut either on that recognition sequence or close to that recognition sequence, right? And they also modify the sequence by producing sticky ends, blunt ends, etc. There is also another restriction enzyme called type 3, a single enzyme having two subunits for recognition and cleavage. Now this type 3 restriction enzyme is made up of two subunits that will recognize the specific sequence and cleave. Cleavage site is 24 to 26 base pair from the recognition site. That means after recognizing the sequence, it cleaves 24 to 26 base pair away from the recognition site, from the restriction site. So this can be used, it is more acceptable compared to the type 1 restriction enzyme, which can cleave 1000 base pair away, right? So type 2 is more preferentially used because they cleave at the same site or close to that site, 1 or 2 base pair away. The last is type 2S restriction enzyme. Cleavage site is up to 20 base pair from the recognition site. Okay. So more preferentially we use type 2 and type 3 restriction enzyme, right? Now comes the restriction recognition sequence. Which sequence they identify, what are the sequence, what is the speciality of the sequence that this restriction endonuclease identify and cleave. Let us see. Now if I say in this way, I think you can able to understand the recognition sequence identified by the type 2 restriction endonuclease enzyme and also by the type 3 restriction endonuclease enzyme, that sequence should have a palindromic in nature with rotational symmetry. What does it means? Palindromic sequence with rotational symmetry, what does it means? In palindromes, the base sequence in the second half of the DNA will be the mirror image of the sequence with its first half. Like for example, I am giving a restriction endonuclease enzyme site known as G double A G C double T C. Now this is a palindromic sequence. If you read from 5 prime to 3 prime direction or from 3 prime to 5 prime direction, it will sound like same. This is palindromic sequence. What does palindromic with rotational symmetry means? In palindromic sequence, the base sequence in the second half is the mirror image of the base sequence in the first half. You see them? It just look like a mirror image. The base sequence in the second half is the mirror image to the base sequence in the first half. But I have said that restriction endonuclease enzyme observe the recognition sequence that have palindromic in nature with rotational symmetry. In case of palindromic sequence with rotational symmetry, the base sequence in the first half of one strand of DNA double helix will be the mirror image of the second half of its complementary DNA strand. Again I repeat, then I will draw so that it will make you clear. The base sequence in the first half of one strand of DNA double helix will be the mirror image of the second half of its complementary DNA strand. So let us see. G double A double T C C double T double A G. So you can see here, if I divide them equally, the base sequence in the first half is the mirror image with the base sequence in the second half of its complementary DNA. So this is the complementary DNA of this one. So the base sequence of the first half of one strand of DNA molecule is the mirror image to the second half of its complementary DNA strand. Similarly here also, the base sequence in the second half of the DNA is the mirror image. It is the first half, second half is the mirror image of the first half of its complementary DNA. So this is your palindromic sequence with rotational symmetry. So by observing this type of palindromic sequence with rotational symmetry, your restriction endonuclease enzyme, specifically type 2 restriction endonuclease enzyme, identify and cleave, means it cut at that particular site. Okay, various type of restriction enzymes are being given here. Let us see. Okay, I think you can able to see, like eco r1. Eco r1 I have already discussed. It is taken from E. coli. This restriction enzyme can identify this sequence G double A double T C and C double T double A G. Just now I have drawn this one where it cut between G and A. If I draw broadly here, G double A double T C, C double T double A G. So it cleaves, eco R1 normally cleaves between G and A in both the strands, both the complementary strands. This is the restriction site of eco R1. Okay. Second, BAM H1. BAM H1 is actually taken from B for bacillus, AM for amylolecu faciens. Bacillus amylolecu faciens. H refers to the strain number and R1 refers to the first discovered restriction enzyme from Bacillus amylolecu faciens. Okay, what is the restriction enzyme sites of BAM H1? Let us see. GGATCC. The opposite complementary strand will be CCTAGG. It cleaves between G and G. both in the complementary strands, and thus it produces sticky ends, right? We can insert our DNA of interest into this site inside, and we can join them. But our DNA of interest should also have that type of sticky ends. Then HAE3. HAE3 taken from Haemophilus aegyptius. Haemophilus aegyptius. H for Haemophilus, AE for aegyptius. 3 refers to the third restriction enzyme which has been discovered. Simple restriction enzyme site it recognized double G double C. Okay, double G double C, double C double G. It cleaves between G and C. It cleaves between G and C and producing blunt ends GGCC CCGG. It producing blunt ends, not sticky ends. Okay, just like this. Then comes hint 3. H for Haemophilus, IN for influenzae, Haemophilus influenzae. D refers to the strain number, and 3 refers to the third restoration enzyme which has been discovered. It actually identifies this sequence double A G C double T, double A G C double T. Its complementary will be double T C G double A, and it cleaves between A and A to the both the strands and producing sticky ends. You can see having overhanging end. Sticky end means one of the strand will have overhanging ends, the other strand will not have. But in blunt end, there is no such overhanging end presence. Is there is any overhanging end? No. That's why it is blunt end. And but in sticky ends, there is a overhanging ends at the extreme position. That is why we call sticky ends. And sticky ends are very easy for us to use because by generating sticky ends we can able to insert our DNA that should also have a sticky end like that and have to join them properly by complementary base pairing. Then another enzyme is there NO okay NOT. Okay, it will be NOT1 because the name of the organism is Nocardia otitis, Nocardia otitis, and it recognized this so much large sequence GCGGCCGC. Only GC it identifies actually, and cleavage occurs between CG in both the strands, thereby producing sticky ends. That is having the overhanging end at extreme point. Now, most of the restriction endonuclease, particularly type 2 restriction endonuclease, cut the DNA at definite sites within the recognition sequence. Selected list of enzymes, their recognition sequence, and their products has been given in the table that I have shown. The cut DNA fragments by restriction endonuclease may mostly have sticky ends, which have overhanging ends, or it may have blunt ends. DNA fragments with sticky ends are useful for recombinant DNA experiments because it helps to ligate very easily, very quickly. Having the sticky ends, having that overhanging ends, it helps the DNA to ligate quickly. This is because single-stranded sticky ends can easily pair with any other DNA fragment having complementary sticky ends. This is mainly important that why DNA fragments with sticky ends are useful for DNA recombinant DNA experiments because the single stranded sticky DNA ends can easily pair with any other DNA fragment having complementary sticky ends. So we can insert our desirable DNA fragment that should have a complementary sticky ends. It can easily binds, and obviously we require ligase, but it is more wellly binded properly. Okay, in the next class I will try to discuss about the DNA ligase. It almostly became 1 hour, so I was trying to go for all the tools that are required for recombinant DNA technology experiments. So we will do the next things like DNA ligase, then your the homopolymer tailing, linkers and adapters, then your alkaline phosphatase, then your other DNA modifying enzymes like nuclease, polymerase. You know the name of polymerase, okay, in the next classes. And then I will try to discuss about the plasmid vectors, and basically I will try to finish off your plant biotechnology class by going directly to that plasmid which is being used for plant biotechnology experiments, that is your TI plasmid and RI plasmid that is given in your syllabus. Okay, so that's all for this much for this lecture, and if you have any doubts regarding this lecture you can ask me in the WhatsApp group. I will try to explain them.